with the synthetic biology group.
So in the synthetic biology group, we looked at a few challenge topics and how they intersect with synthetic biology. For cell mapping, we talked a little about how synthetic DNA libraries could be used for barcoding of cells and cells as they differentiate, and also how synthetic libraries could be used for fluorescent labeling of cells. For gene editing, we talked about how CRISPR is being used for converting one genome into another. So the example we gave was eGenesis, using converting pig organs into human organs, which came up with a number of subsequent challenges, which I'll come on to in a second. Growth factors, we looked at how potentially synthetic biology could be used for creating a synthetic FBS, fetal bovine serum, for mammalian cell culture, that at the moment that's one of the bottlenecks. Certainly we talked about the cellular agriculture industry, the synthetic meat industry, which is currently booming, and one of their challenges that they're trying to solve is how to grow stem cells for meat cheaply. So potentially there will be a new form of FBS that's used for that industry that could potentially make it much easier for this industry, the organ industry, to produce organs. Next we looked at cell sourcing. Can we produce or synthesize synthetic proximal tubular cells for kidney production? And finally, just going back to the issue of converting pig organs into human organs, there's a whole bunch of issues around how to remove the viral load or viruses that are found in pig organs once you transfer them into human organs. That was a big challenge, and the issue of prions or prions. And the last thing that we looked at when we looked at other challenges that could be accelerated, we talked a lot about epigenetics and how there's a huge role of epigenetics in fate mapping, cell mapping, and one of the biggest issues is knowing the unknowns. That the epigenetic landscape is so large that there's so much that's unknown about it. So one of the foundational technologies that we could invest in would be a map for DNA and RNA modifications. And then we talked about the potential for real-time mapping of the transcriptome and a new method called CLIP. So I think the microphone is dying of battery. A new method called CLIP, cross-linking by immunoprecipitation, that could potentially provide real-time mapping of that.
Cryopreservation.
Okay, so how could advancements in each enabling technology potentially accelerate solutions to challenges or sub-challenges? I think in general, the theme that emerged is that depending on what you're talking about doing, it's a really different story. So just preserving ECM, preserving cells that you're sourcing, are they iPSC derived? Is it an allogeneic tissue? Is it something that's homogeneous? Is it something that has an extremely complex architecture? All these things really change not just what you would do, but the level of difficulty involved. And I think the simplest thing at the end of the spectrum was bone marrow, which you can cryopreserve with extremely simple solutions today with 10% DMSO. At the other end, I think you've got vascularized, viable, whole organ, and that's a much bigger challenge. So that brings us to number 2, what technology advancements are needed. To be honest, we didn't really attack that in a direct way, and I think that's because we spent so much time talking about the kind of different, diverse needs. I'll put back on my OPA hat, sorry Josh, not my committee chair hat, and say You can come up to me afterward and I'll give you the link to the Nature Biotechnology article and related articles, which does a really nice job summarizing that. $50,000 prize topic we didn't get to at all. Does anybody have standout ideas they want to just give unfiltered? Sorry I haven't gotten to vet this, guys. Okay, cryoprotective bioinks. All right. Who's this go to? Didn't see you there. Jump in.
All right. Okay, so in the microgravity group, we talked about how we could look at things like simulated microgravity, other work that has been done on station. There's obviously a lot of work that's been done during shuttle days, which were shorter duration experiments, how we could maybe take the literature that's out there, the published literature, and try to segment that into tissue-specific opportunities so that we can really understand how to help each of the individual tissue groups that are working on specific challenges. We also talked a little bit about how microgravity really can be used to answer the question that you're trying to ask. So not just the, hey, it's cool, I'm gonna fly it and see what happens, but really understand how you are able to use that microgravity environment to really inform your research. And we talked a lot about the fact that, you know, the sense of microgravity research being a one-off or very expensive and you get one shot to answer the question, and those days are actually changing quite a bit with the commercialization activities that are going on and sort of more companies stepping in that are doing more research on station. A lot of automated systems, not quite as much crew time, sampling, things like 1G controls on station from centrifuges. So, you know, the ability to handle multiple temperatures up and down and sort of really even remotely control your experiments from the ground. So that if you realize at day 10 that you probably shouldn't wait until day 14 to fix your experiment, you could do that and make sure that you make that adjustment to kind of save your science. So, you know, before I think it was more of a sense of it's a black box, you fly it, you cross your fingers, you hope when it comes back that you've got good data and live cells, but we're getting much better at being able to visualize what's happening during the process, downlink the data remotely, make changes during the experiments on orbit if we need to as well, and really accommodate systems that allow people to automate and think about doing more than just one experiment. So bringing those costs down a little bit too. We talked a little bit about the prizes, but not really. So we didn't have a great chance to do it either. But I think as I mentioned sort of earlier, you know, I think one area you heard the answer about epigenetics and how that's so important in really understanding the whole tissue engineering piece for different types of tissues. And I think we have an opportunity, even as we look at different cell types and we look at the epigenetics, we can add information into that data pool. So it may be as simple as flying cell lines where we just take a look before we go to the, let's turn them into spheroids, let's turn them into pieces of tissue or eventually whole organs. And that may have a significant contribution to the field. So we're excited and hopefully lots more research on station.
Yes, so we've examined two of the topics, vascularization and then patterning and morphogenesis. Those were our focus, since that's the enabling technology committee that we are. So the first topic that we discussed were really what is happening right now with AI and deep learning, which is quite revolutionary in the ophthalmology instrumentation. I'm assuming also in other kinds of medical imaging. So I know from reviewing papers in this area, the extraction of a vascular pattern, essentially a binary vascular pattern from tissues, is a topic IEEE. I mean, It's just a world-class global problem that people have not been able to solve for some decades. But bringing in the new deep learning, AI as we have represented here at Ames, for example, in our supercomputing division, not only this group, but people around the world in that area are now starting to be able to much more efficiently extract these vascular patterns in both 2D and 3D. So definitely for the tissue engineering, the 3D bioprinting, this kind of definition of the vascular architecture for solving the vascularization challenge is clearly fundamental. So we're thinking we would be able to work with the decellularization scaffolds, for example, that have been discussed extensively today. This could in turn inform blueprints for the 3D bioprinting in the future. And some of the issues, the challenges related to that are the fact that we need to have healthy patterns, not just from compromised donors, for example. So that was the first topic, and I think that's really on the verge of making a lot of breakthroughs in the next couple of years around the world, but also within our alliance if we pursue that. Now, then we switched over to talking with the vascular biologists and physicians' perspective, some of what they are looking for from this kind of an enabling technology committee. So again, the definition of the vascular architecture for different tissues, different organs, for the 3D bioprinting, other approaches, that was clearly fundamental. And the kinds of branching roles and approaches that we have our own perspective that we're working with, with the VestGen software is one example. Then another interesting discussion was about how these organs tend to be deterministically determined, the major arteries and veins going in and out. But then inherent for humans and mammals is the idea of also our individual variability. So to what extent do we need to go beyond a very deterministic pattern to creating some kind of variability or randomness? I think this is an open question that is fascinating in biology and medicine for us to think about in terms of truly efficient vascularization. For everyone's retinal vascular pattern, your left eye, your right eye, every person here, each of your retinal patterns is as individual as a snowflake or a fingerprint, absolutely unique identifiers. At the same time, the space-filling properties of these vascular patterns, yours, mine, are very uniform in healthy tissues. The space-filling properties and the oxygen delivery. So that's always fascinated me. Then what are some? Well, we did talk about, actually, for the work that we're doing, if you have any 3D multi-photon data sets, for example, from the mouse ear or other kinds of architectures from these decellularization constructs, we could use more data as, for example, in our own research, and there may be other people interested in sharing these kinds of images. And let's see. Then we talked about some of the questions that always come up in angiogenesis and vascularization, tubulogenesis, sprouting, these different mechanisms, how are they determined? And they're actually different in inflammation compared to normal healthy development and wound healing. So that's another interesting problem, I would say, for the future as we examine the efficiency of different vascularization strategies. So, thank you.
Great, thank you. So thank you to everybody that was able to lead the discussions and just be involved in all of this. So, this was number 3 of our road mapping workshops, right? We started middle of 2015 just to get the idea together. End of 2016, we did a program specifically on the vascular tissue challenge and the side of where's the role of vascularization in all of this. This year is really about refining all of the details of this roadmap. And when I say all of the details, I mean the first round of all of the details, because of course, this is going to be a long-term effort. But what we gathered from these discussions today is going to really help us move forward on refining the roadmap to the point where we can put it out there to the public and start using this as a tool to bring in other resources. I mean, that's what this is all about, right? We want to bring in other resources to help you guys accelerate your research. whether that's other funding opportunities or other expertise. I think this last roundtable session was a lot about other expertise. I mean, we've got experts that I never thought we'd have connecting in with biotechnology. There's a lot of other enabling technologies that we haven't talked about yet. So even if you have friends or colleagues that you think might have a role with other technology areas that could potentially have an enabling aspect on bioengineering, bring them in. Let us talk with them, because I think that'd be something really important to add into the roadmap as we grow and as we go forward. I'll talk more tomorrow about some of the next steps with the New Oregon Alliance and the roadmap and future prize activities. Tomorrow we also have some really great speakers who will be leading us into some other thought-provoking discussion areas. We've got a session about funding and commercialization, and really how do we think about taking science into the business side. We'll hear from folks at NSF, the Methuselah Fund, which is the investment arm of the foundation. We also have an exciting speaker on communicating science, because that's always another big part of what we're doing with the roadmap and everything else. Is we need to be able to communicate all of these challenges out there in order to bring more people in to support us. So communicating science is going to be a really important part of all of this as we move forward. I think the last program that we have for tomorrow is on standardizations. So thinking about, well, how do we standardize everything that's going into this? So we're going to have an expert from that field talk to us about some of the efforts that our group is doing in this. And then we'll close it out with kind of group discussion and a little talk on what are the next steps. That's really what the New Oregon Alliance and all the partners involved in this are focused on, is how do we move forward in actionable next steps to make a difference. That's one of the reasons why we put these $50,000 questions in there, because that might be an actionable next step that we could take relatively quickly. So if we can come up with things that are even incremental opportunities to say, look, this is something that now as a community we know how to do, that's going to mean a lot to then taking the next step after that and the next after that. So that's what we're all about. It's system science for advancing biotechnology and bioengineering. I hope everybody had a great day, and I hope that you guys will have a good evening, relax a little bit, and be back here with us tomorrow morning. I think our program starts at 9 a.m. tomorrow. We'll have breakfast, I think, at 8:30 available for people. So please come back ready to discuss the next steps of where we're going with all of this. And have a great evening. Also, one other point of note, if you filled out your discussion prompts and feel like they're done, please leave them up here for me. If you still want to think about them more, we'll get them from you tomorrow. And if I can see any of our oversight committee members or committee chairs up here just for a few minutes, that would be great. Thank you all. We'll see you tomorrow.